So I'm just going to welcome everyone and thank you for joining us today. My name is Leah Rosen. I'm the online editor for BioProcess International. And before we get started, just a couple of notes. The webcast is being recorded and will be made available for a replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us. It is now my pleasure to introduce our speaker, René Gantier from Repligen.
Hi, thank you, Leah, for the for the introduction, and hello everyone. Very excited to giving this talk today. We'll talk about selecting the right technologies to advance the manufacturing processes for gene therapy to the required level of productivity to meet the demand, meeting the demand now and in the future for gene therapy. At Repligen, we have a concerted initiative on gene therapy, and we want to partner with the industry to provide technology solutions to accelerate the journey from gene to therapy. If we look at the manufacturing challenges to make gene therapy a global success, those are well identified. We need to increase the upstream and the downstream productivity to produce enough doses to meet the demand, and this requires a thorough process optimization and use of scalable technologies. We need to do it quickly to reduce the time to market using high throughput techniques. And of course, we cannot compromise with the product safety and quality. And we can achieve that thanks to the adoption of automated systems and closed processes. So let's consider these issues and solutions one after the other. The current standard process to produce viral vectors, let's call it gene therapy manufacturing 1.0, is often limited in productivity and scalability, coming from the use of adherent cell culture and transient expression, and also results in low downstream yield. So there are some ongoing developments related to cell lines, media and process optimizations that will lead us, hopefully in the next 2-3 years, to the gene therapy manufacturing 2.0, with producer cell lines grown in suspension mode at high cell density to produce high virus titers, and optimized purification processes to prevent virus loss and or inactivation. So it's not surprising to see most of the gene therapy companies now adopting suspension cell culture for the development of their new treatments. This data here is just one example published about a successful case of AAV vectors produced from HEC293 cells adapted to suspension. And this is showing an increased overall process productivity coming from the suspension cell culture. So the transition from adherent to suspension mode adds safety to the process with no animal-derived material used, and also significantly facilitates the scale-up of the process. But the titer in suspension mode is generally lower compared to that of obtaining adherent mode. So even if the transition to suspension is beneficial for safety and scalability reasons, some further improvement is required to increase the titer. And this can be achieved by intensifying the process, going from batch to perfusion cell culture. Here is such an example coming from one of our customers, Cytovance, using the Repligen XL ATF technology, which is a cell retention technique that is enabling very high cell density in perfusion mode. It's broadly adopted already for the production of recombinant proteins and mAbs, monoclonal antibodies, and now it's also implemented for gene therapy processes. So here, the productivity of optimized XL-ATF perfusion runs was compared to that of batch cell culture. The XL-ATF perfusion process enabled a higher cell density before transfection. And this, combined with some cell culture optimization, led to a higher than 100-fold capsid productivity increase. So this is one example for high productivity with AAV in perfusion mode. What about lentivirus? Well, we can do high productivity processes for lentivirus too, thanks to the new Repligen TFDF technology. So the TFDF technique is uses a combination of tangential and depth filtration techniques in a single filter. So it enables the cell retention process while harvesting the product without fouling the filter. So this example here was recently published by Oxford Biomedica and Repligen, and it shows how the TFDF technique was applied for high productivity harvest of lentivirus. The suspension cell culture was performed in batch mode, but then on one hand, a standard harvest using depth filtration was conducted, while on the other hand, the harvest was done with the TFDF technique. The depth filtration process, as you can see, led to about 70% yield, while the TFDF harvest was at 100%. But more importantly, the TFDF harvest kept the cells intact in the bioreactor. So after having replenished the bioreactor with fresh media, the cells continued to produce the virus, and a second harvest was possible, leading to a total yield of about 200% compared to the depth filtration process. So I believe these two examples with AAV and lentivirus demonstrate that we are technology ready to overcome the upstream productivity challenge, thanks to the adoption of suspension cell culture and intensified perfusion processes. We can go even further if we think about the new producer cell lines coming. We will be able to use the same tools for these new cells. So that's really encouraging. And as we said, looking at the downstream side of the process now, what are the challenges on that side? We see a cumulative effect during the downstream purification of viral vectors, lowering the yield step after step, and sometimes resulting in the loss of about 90% of the active product. Most of the loss is coming from the two common chromatography steps with non-optimized separation conditions, leading to compromise between yield and purity, but also from harsh conditions that can be used in terms of pH, connectivity, etc., leading to virus inactivation. Additionally, non-optimized TFF steps will also lead to significant product losses due to shear forces, the product being bound onto the membrane, or simply loss in the older volume because of wrong sizing of the system. So it's therefore critical to use the right technology during the process development to optimize this process. So we are talking about the challenges on the downstream side, and we talked about the chromatography. How to improve the process on the chromatography side. So on this side, the use of small volume high throughput screening tools will help you rapidly identifying the best separation conditions among a huge number of potential combination between resin types, flow rate, binding capacities, etc. And then the identified process can then rapidly be scaled up using the corresponding scale-up technology. So at Repligen, we offer such a complete tool set that is called Opus Prepack Columns, with a comprehensive choice of chromatography resins, chemistries that are prepacked in different formats and scales. So it starts with the RoboColumns that you can see on the left-hand side, and those are used for rapid and high throughput screening of the separation conditions before fine-tuning this condition to a scalable process with the minichrome prepack column, then validate the process with the valichrome columns before finally scaling up to the process scale. So thanks to these tools, you really optimize and scale up your chromatography process steps. So having done this, having done the chromatography part, let's improve the TFF performance. So the first thing to consider here is selecting the best format between hollow fibers and flat sheet cassettes, which will generate more or less shear-related stress. So if your product is sensitive to shear, like lentivirus, the hollow fiber format is better suited. If you have less shear-sensitive vectors, like AAV, the TangenX flat sheet cassettes provide a good option. So this graph is showing how the higher shear generated with the TFF cassettes compared to hollow fibers can impact the yield, with typically only about 50% of infectious particles recovered compared to almost full recovery with hollow fibers. So you can still test both formats, but we would strongly recommend the use of hollow fibers for lentivirus applications. And then the next step, having selected the format, is, and again, here it's hollow fibers, but it could be a flat sheet cassette. So having selected the format, it's important to evaluate multiple molecular weight cutoff membranes to first isolate the one with the best yield, but also contributing to the purification by removing impurities. So in this case here, we don't see a clear difference in terms of recovery of infectious particles, but the highest molecular weight cutoff is showing better HCP removal and would be considered and selected for the next step. So having done this, finally, we'll go for some flux optimization that will decide the process conditions. And very importantly from there, we will guide you step by step to define your process scale-up with filter sizing, operating conditions, selecting the automated hardware corresponding to each scale to run really your process on the optimum conditions, minimizing the human intervention to save handling time, but also minimizing the risk of human errors. And that brings us to the last challenge to consider for gene therapy manufacturing, which is securing the product quality and safety by implementing closed processing. We can definitely help securing the process by providing you complete and integrated single-use flow path for each unit operation. But even further, we want to give you the opportunity to get the flow path customized to your own process. This flow path would be is delivered pre-assembled, gamma irradiated, and ready to connect via sterile connections. So that makes really a further strong step towards safe processing for gene therapy manufacturing. So, starting with the gene therapy manufacturing 1.0, going through technology-driven process optimization and innovation, we are now coming closer to that gene therapy manufacturing 2.0, where the viral vectors will be continuously produced from producer cells using intensified suspension cell culture run in perfusion mode and purified through a secured and high-efficiency downstream process. As I said at the beginning of this presentation, we want to partner with the gene therapy industry, to accelerate this journey. And for this, we have a dedicated team of experts in the field that are ready to engage the conversation with you to understand your specific challenges and provide technology solutions to overcome these challenges. So with this, I thank you very much for your attention. Hopefully we'll have a few more minutes to take questions. But anyway, feel free to reach out to me. You can see my email here. And I would be very happy to answer whatever question you have in terms of further information that you would need on our technologies for gene therapy. Thanks again.
So the first question from the audience we have is, how are aggregation issues resolved?
Aggregation of the vectors, the virus aggregating. So it depends at which step we are talking about. So most of the aggregation that we can see is during the tangential flow, the concentration of the product, and mostly at the very end of the process where the concentration is the highest. The way to mitigate that aggregation, and it depends on the vectors that we are using. AAV is well known for being prone to aggregation. So this one, to prevent the aggregation, you will have to select really carefully of course, the conditions that you use during the concentration in terms of flow, in terms of pressure, etc. But also, I think it's even more important to select the right buffers, having the right connectivity to prevent such an aggregation.
And one person says they have tried to increase host cell density without observing a significant viral count. What experience have you had with this?
So can you repeat the question, please?
I've tried to increase host cell density without observing a significant difference in viral count. What experience have you had with this?
Okay. So yeah, unfortunately, the biology of viral vectors is not completely and well understood. So yes, increasing the cell density will not necessarily lead to a higher number of virus produced. So there's a combination of optimization to be made as you increase the cell density. That is what I showed in the example of cytovents. There was a combination of increasing the cell density plus some cell culture optimization and transfection condition as well. That plays a big role in the possibility really to get the full benefit in terms of virus productivity from the increased cell density.
Okay, great. And what tool and technique are you using for high flow through transfection?
For the high flow through the transfection, this is
high flow through transfection, or for flow through transfection?
For flow through transfection, I'm not sure I understand the question. So we don't provide any specific technology for transfection. The current, for instance, perfusion processes like ATF will not require necessarily to stop the process before or after transfection to do that transfection. It's just most of the transfection reagent now can be used without doing a media exchange. So it's possible to do that with our technology. But if it requires a media exchange, we can also use the ATF perfusion mode. I'm not sure it answers the question, but that is the way I see it.
Okay, great. Well, I think we have time for just one more question. If we don't get to your question or didn't get to your question, we'll be sending your questions and your information to René, and he can follow up with you directly. So thanks again for all these great questions, and go ahead and continue to ask questions. We will be passing those on. So the last question we're going to ask is, what are the expectations for viral clearance in downstream? Is it mainly from chromatography steps?
No, I mean, yes, there will be some contribution to that, as it is for the monoclonal antibody. We can take advantage of any low pH condition within the process, like it is the case after the affinity step for the AAV purification. There will be some further viral clearance on the second step. But yeah, the issue of the complete viral safety is an open question for viral vectors. And how can we do viral clearance when the product is a virus? It's a challenge. But yeah, some developments are ongoing in that side. Yeah.
Okay. Well, thank you, René.
Thank you. Thank you very much.
Thanks. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements on your inbox. Goodbye.
Bye-bye. Thank you again.